This underground vault on the Norwegian island of Spitsbergen, inside the Arctic Circle, stores millions of the world's most important agricultural seeds. Each seed stored here represents the collective labor and research of hundreds of generations of farmers and gatherers over the course of thousands of years. The seeds are kept in airtight bags, in boxes within larger boxes, deep inside the vault. The Svalbard Global Seed Vault is an attempt to protect the diversity of the world's food crops from environmental or cultural disaster. During the tumultuous period at the turn of the second millennium, several regional seed banks were lost due to wars and commercial expansion. Today, some address this concern by sending copies of their seed collections to the Svalbard Global Seed Vault. Seed banks that deposit at Svalbard are encouraged to replenish their stock every 6 years to ensure its viability. At the entrance to the vault, a diesel generator supplies power to an air conditioning unit that maintains the internal temperature at negative 18 degrees Celsius in accordance with standard seed preservation practices.
I'm the director of the Center for Post-Natural History, which, if you haven't been upstairs yet, we've got an exhibit up there of specimens of organisms that we consider to be post-natural that were produced here in the European Union. Essentially, we started collecting specimens of organisms 5 or 6 years ago that initially we were focused on just organisms that had been genetically modified, and we sort of grew over time to include selectively bred organisms, domesticated organisms, really everything that had been like physically altered by people in a way that was kind of bred really into its DNA, which is really what our definition of post-natural comes down to. It's the living things that have been intentionally altered by people. It's stuff we do on purpose, and it's heritable. It has, you might say, evolutionary consequence. So, where does that start? Where does something become post-natural? Well, part of it is when you take an organism out of its habitat and you bring it under control of like a human habitat. You start raising it in captivity. And when we do that, when we start to take responsibility for its habitat, the organism starts to change. We alter, what you might call the selection pressures on that organism, and we have new ones. We give it, all the food that it wants of a particular kind, or all these kinds of choices we make affect the organism, how it shapes, the shape that it takes. But we also, when we do that, start raising it outside of its habitat, we take responsibility for its reproductive life. We actually insert ourselves into the sex life of that organism in a very real, direct kind of way. We take responsibility for what the bees do with plants. We get involved with both the male and the female sides of the sexual equation, harvesting sperm, eggs, fertilizing them. We become an essential part of the reproductive life of that organism in all the graphic sorts of ways that you might imagine. You can see the guy that's working with the pig female sow on the bottom there. Many times we're like imitating what the animal might be experiencing during the sex act. And this happens at every scale. I believe that's a goose penis that's just erupting there on the lower right-hand side, and all the way up to the scale of elephants and zoos or endangered organisms whose habitat may no longer even exist. We are completely responsible for its habitat. It's worth remembering, I won't belabor the point in this particular talk, but there was a long period of time when we were also approaching humans with the same mindset. The concept of eugenics, which had a very popular period in the late 19th, early 20th century, where we were essentially taking responsibility, the state was taking responsibility for humans' sex lives as well, promoting certain people to reproduce together and trying to prevent certain kinds of other people from reproducing together. So this is the side of the equation that I want to talk about a little bit more is all these different strategies where corporations, governments, laboratories use to control living things from reproducing outside of the space of the lab, of containment for all different sorts of reasons. The most kind of obvious and earliest to be developed was essentially castration, or removal of the sex organs would kind of prevent this organism from reproducing. So we still do this to our pets and farm animals, depending. But when we get into the era of biotechnology, we start to develop a proliferation of strategies to address the problem of reproduction, a problem of life making more of itself for free. So the first one I want to talk about addresses this problem in the early days of genetic engineering. So in the mid-70s, when we were first taking DNA from one kind of species and putting it to another, and this is happening in bacteria, very smallest organisms, there was a lot of concern about what would happen if these were to get out. There's a meeting called a Silomar, where scientists actually got together and kind of were brainstorming this. And for a while, it was like they called timeout, and everybody sort of paused their research. And one of the strategies, the ideas for containment that came out of that was, what if we created a new cell line, a new kind of E. coli bacteria that can't live outside of the lab? It requires some essential nutrients that we supply it in the lab that it can't get in the wild. And they called this, in honor of the American bicentennial, E. coli 1776. And this was used, if you look in the literature, it seemed to have kind of a period of use that sort of waned as the 80s continued on. Apparently it was very difficult to work with and was subsequently abandoned. At this point, it may only exist in some freezers somewhere in a lab. That may be the last remaining habitat of E. coli 1776. It may actually be considered to be extinct at this point. When life becomes a property, not just a tool of research, but something that is to be bought and sold, an additional kind of problem comes up with this reproductive business. And that is to say that when your product makes more of itself for free, selling it for money becomes a problem. So this is specific to capitalism here. So one of the ways that companies have addressed this is through contractual agreements. So Monsanto, quite famously, is very protective of their seed product and has produced quite an elaborate contractual agreement. This is very similar to what you agree to when you open a pack of software, for example. This is called the Monsanto Technology Stewardship Agreement. So if you just parse out the title, the technology that they're talking about is corn or soybean seeds, and the steward is the farmer. The reason they call him a steward is they're not selling him the seeds. He's taking care of it for them. But this agreement says that they still own all of it. The farmer assumes all the responsibility for it, and the farmer has to follow a lot of instructions that basically relate to the containment of the organism. So the farmer's not allowed to keep the seed, not allowed to replant the seed, not allowed to resell the seed, or give it away, and they have to douse the seed in lots and lots of Monsanto's main product line, the Roundup herbicide that the corn has been designed to be resistant to. Also similar to the software agreements, these terms are agreed to by simply opening the bag of seed. So you don't have to actually sign the papers. By opening the bag, you've already agreed to whatever that was just said there. Upstairs, you'll see we have a small vial of Roundup Ready corn. And if you listen to the narrative that accompanies it, it describes the kind of lengthy steps that we had to take to get this corn without violating those terms of conditions that just went by. So for each of the strategies that I'm describing, there are counter strategies for whatever reason where people would like to see a particular organism reproduced. Another strategy that plays out in the commercial world, we could call it security through obscurity, is to not describe, unless you absolutely have to, the nature of the organism that is being genetically engineered. So in the United States, every time an organism leaves the lab, you have to file for a particular kind of release permit. I think there's a similar system in the EU. And this is a matter of public record. So there's a large database. This is one entry of very many. This was a particular kind of corn. It's assigned a number. It's issued to Monsanto. And then there's this long list of genes that might be in the corn. Usually there's just a couple in these entries. This one, they kind of went for broke, I guess, and kind of got the largest permit they could. And it's for specifically Hawaii and Puerto Rico. So very site-specific. But you'll see islands, which that's a different talk. But you'll see each entry has little CBI next to the name. And the CBI is blotting out what would be the name of the gene. So in this case, CBI stands for confidential business information. So this is the big black marker that would be kind of erasing that name. As you go through the database, sometimes you'll see CBI listed as the species itself. Sometimes it's all CBI, and you don't really know anything about it, and we're essentially left to conjecture as to what this organism is. What I found kind of compelling was that within the database, you could search for a species, and they had a long list of all the different species that are in this. So you had alfalfa, soy, corn, all these familiar organisms, and then CBI was listed as an organism. So at our museum in the US, we always have CBI as one of the organisms on display. It's essentially the organism of conjecture. It's the one we don't know anything about, and therefore we're free to imagine whatever we like about it. The last group of strategies that I'm going to talk about are where the reproductive control is built into the DNA of the organism. So 1998, Delta and Pine Company patented a system that kind of subsequently was called the Terminator gene. Monsanto purchased it. This was essentially a genetic mechanism whereby the organism would not, you could essentially turn off its ability to reproduce through the presence of an antibiotic, tetracycline. So within their system, they could make more of this stuff, and when it leaves the system, it would essentially, its next generation would be unable to reproduce. Essentially, I think when they reach a certain stage of early development, it triggers a kind of a suicide of the seed. This was met with kind of worldwide controversy. It's important to remember that when they first proposed this system, the idea was containment, was to address the concerns that these crops would escape into the wild, contaminate other crops. So this way, they created a system whereby it could not breed. But this also had another consequence for farmers all over the world, where there is a tradition of replanting your seed every year, choosing the best crops. Essentially, if they were to allow this seed into their local seed economy, they would be dependent on Monsanto. They would essentially be subscribing to every year to a new batch of seed and be entirely dependent on this company. So very, very controversial system. So controversial that it never got implemented. We talk about it a lot. People protested all over the world. No seed made it to the market with the Terminator gene intact. Yet every company that makes genetically modified crops has their own patent on their own kind of version of this system. So it's still desired. Where we have seen it is, and this is the last organism story I'll tell, is a genetically modified mosquito. These are being tested on a number of islands. I always like to test on islands first. By a company called Oxitec, and they basically market pests. Mosquitoes, Aedes aegypti bullworm that eats corn in the US, Mex fly, species that carry disease that affect people or eat foods that affect people. And what they market is a version of that pest that they have reproductive control over. so that they could grow up tens of thousands, hundreds of thousands of this organism, and then essentially disable their ability to reproduce and release them into the wild. And in doing that, they ineffectually mate with their wild counterparts, and no offspring are produced, and each generation the population kind of goes down and goes down and goes down, and you keep repeating this until that species is disappeared. This was successfully tried in the US with the screwworm, and so they're kind of repeating that strategy now using genetic engineering instead of with the screwworm. This is the 50s, so they used atomic radiation. But it's interesting to see that the terminator gene has kind of found its way out of the lab with a lot less controversy now. It's not being paid attention to quite as closely. So I just wanted to put those kinds of strategies out there, that we've got ones that exist on paper, these contractual agreements, ones that are built into the DNA of an organism, and sometimes where we're altering the organism itself. There's different ways that kind of control comes into play with these kinds of organisms.
Any questions from the audience in the meantime?
Yes, I want to thank you for the presentation. It was very interesting. And my question is, in your practice, what kind of ethical guidelines do you follow, and how does it work, and how this environment changes now? Because there are a lot, of course, happening now in this sphere of technology and biotechnologies, and I know that there is more attention is paid in academia also to the ethical side of it, and maybe you can tell a little bit about it.
I don't do the lab work. I don't make these organisms. I primarily exist as a documentarian. So my ethics are to keep things in public as much as possible, so to add a certain degree of transparency, but also to tell stories that are fair. All right, that fairness would be an ethical concern of mine, in an effort to allow people that have differences of opinion to kind of coexist, to have a conversation, to have a dialogue around it. I guess that is kind of an ethical position to take, that actually suspending my own internal sense of morality and ethics in favor of a kind of public neutrality, that's an important role. I'm not saying everybody should take that position, but that was a need that I saw, that there was all this kind of conversation happening around these issues, but there was no place where you could kind of go just to learn about it, to meditate on it, to come to your own conclusion. There's kind of a lot of voices trying to convince people to believe one thing or another. So I guess my ethical position has been to suspend my own ethics verbally in favor of allowing other people to get louder about their ethical considerations. I try to tell stories that are different from maybe how these issues are typically framed, because I want people to kind of hear them for the first time. So oftentimes, as you'll hear, like when in the exhibits, we kind of find an unexpected way of talking about an organism that doesn't just hit these kind of common, kind of already well-marked political kind of battlefields. So if I hear a narrative coming up kind of very regularly, it's well understood, then that's a story I don't need to tell. So it does. And if I hear a narrative that I haven't heard before, that may very well become a significant part of an exhibit. So it's adaptive.
Thank you.
Genetically modified foods are a source of great controversy in the European Union. Therefore, most are only approved for use in animal feed, or must be processed into basic starches, sugars, or oils before they may be eaten by humans. Typically, these plants are modified in two ways. First, they are made to be resistant to one or more patented herbicides that are sold by the same company that produces the seed. Secondly, they are altered to produce a poison that kills insects that may feed on the plant. Upon opening the bag of seed, farmers enter into a license agreement with the manufacturer, similar to agreements that users of computer software must agree to. The license agreement specifies that the company selling the seed remains the sole owner of the seed, and that any negative consequences resulting from the use of the seed is the sole responsibility of the farmer. The contract also requires the users of the seed to douse their crops with the corresponding herbicide on a predetermined schedule. This is intended to extend the effective life of the product until weeds eventually become resistant to the chemical and farmers are compelled to invest in a new product line.